Ladies and gentlemen, it is with great pleasure and immense honor that I am here today to share my research findings in the field of cognitive neurosciences. My name is Yan Li from Xi'an Jiaotong University, People's Republic of China. Today, I am thrilled to present to you my most recent study in the field of neurology, which is dental stem cell-derived extracellular vesicles transfer microRNA 3305P to treat traumatic brain injury by regulating microglia polarization. Mesenchymal stem cells are a type of multipotent adult stem cells that exists in various types of tissues, including bone marrow, umbilical cord, adipose tissue, and others. MSCs possesses strong self-renewal and multilineage differentiation abilities, and they can differentiate into various organs and tissues. Therefore, they have demonstrated significant advantages and potential in the treatment of various diseases. However, the mechanism of MSC therapy for diseases has long been a subject of debate. Early theories suggested that stem cells could treat diseases by migrating to the injury site after injection and further differentiating into damaged tissues and organs. However, this viewpoint was soon challenged after researchers tracked fluorescently labeled stem cells in vivo and found that the survival rate of implanted stem cells in disease-induced microenvironment was extremely low, with less than 1% of cells being able to migrate to the injury site and exert their effects. Consequently, the possibility of these cells directly differentiating into damaged tissues and organs to function was considered unlikely. As a result, a new perspective emerged, suggesting that after transplantation, stem cells secrete therapeutic substances that can impact the injury organs and tissues, promoting repair and regeneration. This phenomenon is referred to as intercellular communication. The extracellular vesicles, which we are about to introduce, played a crucial role in mediating this so-called intercellular communication. Extracellular vesicles are nano-sized membrane-bound vesicles secreted by cells. From this magnified image, we can observe that EVS carried various bioactive substances, including proteins, nucleic acids, lipids, and more. They can enter recipient cells through a different mechanism of action, such as the endocytosis, phagocytosis, and direct fusion, thereby regulating the functions of the recipient cells. In our previous work, we have extracted the 5 most commonly used type of stem cell-derived extracellular vesicles for clinical treatment, which originate from bone marrow, umbilical cord, adipose tissue, dermis, and dental pump. We found that dental stem cell-derived extracellular vesicles contain a higher concentration of neurodegenerative factors. Furthermore, upon intravenous injection, they tend to distribute more toward neuronal tissues. These findings suggested that dental stem cell-derived extracellular vesicles may possess the potential for neural repair. Traumatic brain injury is a significant public health issue that can result from various cases, such as falls, motor vehicle accidents, support injuries, and violence. It is characterized by damage to the brain caused by an external force, leading to a wide range of physical, cognitive, emotional, and behavior impairments. TBI affects millions of people worldwide each year with varying rates across different populations and regions. Males have a higher incidence of TBI compared to females, and the highest rates are typically observed among young adults and older adults. The pathology of traumatic brain injury involves the primary and secondary injuries. Primary injury refers to the immediate damage caused by the initial traumatic event, and secondary events mechanisms occur over time following the primary injury and involve complex cellular and molecular processes. These processes can include inflammation, oxidative stress, mitochondrial dysfunction, and alterations in blood-brain barrier integrity. Secondary injuries can exacerbate brain damage and contribute to the progression of TBI-related complications. These consequences of TBI can be diverse and long-lasting. The severity and outcome of traumatic brain injury can vary widely, ranging from mild cases with temporary symptoms to severe cases with permanent disability or even death. To review the therapeutic potential of dental stem cell-derived extracellular vesicles in treating traumatic brain injury, we selected stem cells from human exfoliated deciduous teeth. We named that SHAD to investigate the reparative effects of the EVS secret on TBI. Behavioral results demonstrated that SHAD EVS significantly restore the impaired motor function in rats. In vitro studies demonstrated that SHAD EVS primarily exert their therapeutic effect in treating TBI by modulating the population of microglial cells to suppress neuroinflammation. After identifying the role of SHAD EVS in modulating microglial cells to treat traumatic brain injury, we sought to elucidate the specific molecular mechanisms underlying their therapeutic effects. Through the high-throughput analysis, we identified the key therapeutic molecule carried by SHAD EVS, which is microRNA 3305P. Functional experiments confirmed the regulatory role of microRNA 3305P in polarization of microglial cells. Next, through the bioinformatics analysis and molecular biology validation, we further identified the downstream target gene of microRNA 3305P as EHMT2 and confirmed their direct binding interaction. Through the Western blotting and chip, we elucidated the downstream signaling pathways of microRNA 3305P and EHMT2. We confirmed that 3305P and EHMT2 regulates the transcription of CXCL14 by modulating H3K9 trimethylation, thereby inhibiting M1 polarization and promoting M2 polarization of microglial cells. Additionally, we also validated our findings in the in vivo level. With a clear understanding of the molecular mechanism underlying the therapeutic effects of SHAD EVS in traumatic brain injury, our next step is to focus on the practical applications and advance the clinical translation of EV-based therapies using SHAD. In previous studies, we observed that when SHAD EVS were locally applied, they didn't stably remain in the injury site and their release was transient. However, to achieve optimal therapeutic effects, it is necessary for SHAD EVS to persist at the injury site and slowly release their contents. To address this issue, we have designed a temperature-sensitive delivery system for SHAD EVS, which respond to the reactive oxygen species, which is ROS, generated in TBI. After local administration, this system allows for slow and sustained release of SHAD EVS at the injury site, with a duration of efficacy lasting up to 21 days. The antioxidant performance of the delivery system was evaluated by detecting the ability to scavenge free radicals. The long-term release profiles of SHAD-EVS was also calculated. In vitro results demonstrated that our SHAD-EVS delivery system can inhibit M1 polarization of microglial cells while promoting M2 polarization, thereby suppressing the neuroinflammation. Through the in vivo experiments, we further confirmed the delivery system of SHAD-EVS in our system yields superior therapeutic effects and compared to direct local injection of SHAD-EVS. In summary, we found that microRNA-3305P transferred from dental stem cell-derived extracellular vesicles rescued the impairment of motor function resulting from TBI. microRNA-3305P shifted microglial polarization by targeting EHMT2, H3K9 trimethylation-mediated CXCL14 transcription, leading to mitigated neuroinflammation and enhanced the repair of brain injury. The engineered dental stem cell-derived extracellular vesicles may be promising in achieving satisfactory functional recovery in TBI and other related neurological disorders. In the end, I would like to express my sincere gratitude to Xi'an Jiaotong University and School of Stomatology, Wuhan University for their invaluable support and collaboration throughout this research project. I also extend my deepest appreciation to all the contributors involved in this research. Their dictation, expertise, and hard work have been essential in conducting experiments, analyzing data, and drawing the meaningful conclusion. Their commitment to scientific excellence has significantly enriched our findings. Furthermore, I would like to acknowledge the generous support of our sponsors. In the end, thank you for your attention also. Thank you so much.